Welcome to the Biological Life Sciences channel. Today we are going to see the technique of Southern blotting and its details. So let's begin. Let's see the history first. The Southern blotting technique was first time discovered by scientist Edwin Southern, and it is one of the important technique till date, and it has no replacement till now. Okay, so it was introduced as a technique to detect particular sequence of DNA in DNA samples, and it was published in first 1975. So it is innovation. It integrates three different techniques. First is restriction digestion using restriction endonucleases, then the gel electrophoresis of that, and then blotting onto a particular membrane. So DNA of different sizes is first of all, what is DNA is taken, intact DNA is taken, it is subjected to restriction digestion by different restriction enzymes, and then this restricted enzymes is now electrophoresed onto agarose gel. Now whatever the bands that are formed on agarose gel, this is blotted onto a membrane through capillary action or through electro blotting assembly, and it can be then subjected for detection, that is hybridization. So let's see the definition. It is an example of RFLP. Southern blotting can be defined as an analytical technique for identifying species-specific sequences of DNA by separating the fragments on a gel and transferring them to a second medium, that can be a carrier medium such as nylon membrane, nitrocellulose membrane, etc., on which the hybridization testing may be carried out. So what will happen here, whatever the bands that are being formed, suppose the bands is having a target of interest or a sequence of interest, then we can detect that sequence using the hybridization method using different probes that can be radio labeled or non-radio labeled. Okay, the DNA is then exposed to hybridization analysis using the probe. There are different types of membrane that are used here, or carrier membrane, and there are transfer buffers, transfer methods to set up the Southern blot assembly. The most commonly and popularly used membranes are nitrocellulose membranes, uncharged nylon positive, charged nylon positive membranes, and it can be interchanged depending on the applications. So let's see the principle. It is based on the principle of transfer of separated DNA in agarose gel electrophoresis to a carrier membrane, which can be a nylon membrane or a nitrocellulose membrane, using gel electrophoresis, and then you can go for the identification of a particular fragment of interest by using the hybridization using different probes. So the probes can be labeled here by using either radioactive detection or a non-radioactive detection. Okay, so DNA is formed between the first of all converted into single strand, and then it is blotted onto a this membrane or a carrier membrane and subjected for detection. So this is the principle behind this technique. So let's see the procedure first in a very short and step-by-step manner. First procedure is to extract and purify the DNA from the cells. The DNA which you want to check or which you want to detect, or the gene which you want to detect whether it is present on a particular cell or a tissue or not, first of all you have to go and extract the DNA sample from the tissue and purify it. Okay, so after purification, then what will happen, the specimen will be incubated with detergents to promote cell lysis, and then all the other proteins and other components of the cell are removed and DNA is purified, and this DNA is then suspended into the buffer and subjected for further experimentation. After this, the fragmentation of DNA is done. This fragmentation of DNA is done by using certain endonucleases enzymes that cuts the DNA into some fragments. Okay, so each restriction enzymes are validated with different kinds of universal buffers which are required for their action. Okay, so appropriate DNA concentration has to be maintained here while doing the digestion, and all time it has to be placed on ice, that is 4 degree centigrade temperature or 0 degree centigrade temperature. After that, you can either go for PCR amplification of this fragment, or you can directly subject it for further analysis if the DNA sample is present in ample amount. Then the next step is gel electrophoresis. Whatever the digested sample of the DNA is, you have to go for the gel electrophoresis here. So the percentage and size of the gel must be determined depending on the molecular weight of the DNA that you are using. The gel consists of microscopic pores and are solid. Usually agarose or polyacrylamide gel is used here. Generally the concentration of agarose or polyacrylamide here is 0.722%. It is considered adequate for most applications. Generally it is 1%. Nucleic acid have negative charge and it moves from left to right, that is positive negative to positive terminal. As nucleic acid is negatively charged, it will migrate towards the positive charge. Okay, so there will be separation based on charge as well as molecular weight. So there will be a banding pattern that will be formed, that can be stained using ethidium bromide to see the banding pattern. Okay, so whenever you are doing the electrophoresis, it is advised to do it in two different wells, so that one well out of that will be considered or will be used for electroblotting purpose, and other well will be taken as a standard sample to compare both the membrane as well as the banding pattern in the first well. Okay, so you can detect the band of interest present in the restricted digested sample inside the lanes. So after that, the denaturation of the DNA is done. Since the DNA is in double stranded form, it has to be denatured first. So alkalis are used to denature, and rehybridization should be prevented. We use NaCl, which neutralizes the DNA here. Okay, after that, the process is taking place is blotting. This blotting is the transfer of DNA from gel to the solid support or carrier membrane. It is done in a capillary action way, or it can be done in a electro-transfer assembly, which is prepared nowadays. We can dry blot also around 80 degrees Celsius using the specific techniques or specific instrumentation required. So generally, electroblotting assembly is done, electroblotting is done after the electrophoresis. If you are using the electroblotting procedure, it takes around 2 to 3 hours. But if you are using a capillary action electroblotting, it can take 12 to 24 hours. Let's see what is hybridization. Once the DNA bands that are blotted onto the membrane, it has to be UV cross-linked to immobilize them, because the membrane will be treated with subsequent buffers, so the DNA must not be immobilized. So for that purpose, different buffers are added on this membrane. So first of all, UV cross-linking is done so that the DNA immobilizes onto the membrane, and after the UV cross-linking, the membrane is baked at 90 degrees centigrade. So this what will happen due to this? The DNA will be immobilized permanently onto the membrane. Okay, so after that, DNA is incubated with a labeled probe. It usually requires 1 to 16 hours depending on the complexity of the probe and concentration. The probe will hybridize at the required point onto the different banding pattern onto the membrane. Then you can detect the probe by using the non-radio labeled detection, or you can detect the probe using radio labeled detection. Okay, so generally the non-radio labeled detection is preferred here, but if radio labeled detection is done, then it can be subjected for autoradiography, and autoradiogram can be generated through which the band can be detected. But in non-radio labeled detection, what will happen? You can use different systems of non-radio labeled blotting. Generally, the system which is used here is biotin related probe. We know that the biotin and streptavidin is having quite an affinity. So the probe is first of all labeled with biotin, and then this biotin related probe is hybridized, and after that there is washing is done to remove extra probe there, and blocking agent blocks the other side on the membrane present. Okay, so after washing with the detergents and NaCl, the membrane is prepared for the action of next, that is streptavidin HRP conjugate. So what will happen? This streptavidin HRP conjugate will bind with biotin, and the streptavidin is having the enzyme HRP, which is a horse radish peroxidase enzyme. So you have to use a substrate that is a chromogenic substrate such as TMB-H2O2. So what will happen when the TMB-H2O2 is added into the medium? The HRP enzyme will act on that, and it will be converted into TMBO. Now this TMBO is giving the blue color to us. So the blue color will be formed at the band where the band of interest or target to be detected is present, and thereby it can be compared with the parent or the second lane that we have electrophoresed, and you can purify the band of interest from that gel using silica gel extraction method. Okay, other way around, you can go for autoradiography. What will happen? The radioactive probe you have used here, or a fluorescent probe you have used here. So directly, if you subject this gel for the fluorescence probe detection or radioactive under X-ray film, what will happen? The probe will give you the fluorescence, and then you can detect the different required band. Okay, you can see this is the traditional capillary way through which the blotting is done. First of all, a wet sponge is taken and buffer is added into this, the electrotransfer buffer, which is specifically required for this purpose, and then there will be gel placed onto that, and then soaked filter paper will be placed below the gel, and then over that the membrane will be placed, that is nitrocellulose membrane, and then it will be placed again with the paper towel, and then a weight is placed onto that. So what will happen, due to the capillary action, whatever the buffer is there, that will penetrate into the gel and into the membrane, and it will take away the bands along with that, and it will blot the bands onto the membrane. Okay, so this takes around this takes a larger time, or it takes around 12 to 24 hours if you do it in this way. But if you are doing an electrotransfer assembly where there will be charges on both sides, generally there will be a positive charge below, negative charge below and positive charge on the top. What will happen, it will extract the DNA onto the positive side and it will be forced to blot on the membrane. So this happens in 2 to 3 hours time. Okay, so after the blotting is done, the membrane is carefully removed and membrane will be having all the band of interest, and if you are doing the radioactive detection, it will directly subjected for autoradiography, X-ray film exposed, and then you can get the band of interest that you want to detect. So this will be a autoradiogram that will be required for detection purpose. So these are the two ways through which the hybridization detection can be done. So there are different applications of this method. So let's discuss the applications. It helps to identify specific DNA from a DNA sample. Identification of viral and bacterial infection also can be done. It is important to study the gene mutation, deletion and rearrangement. It is very important in DNA fingerprinting also, where maternity and paternity analysis and disputes can be solved in forensic studies and personal identification. Diagnosis of neonatal and genetic disorder is also, including cancer, is also done with this technique. Discovery of RFLP is also helpful in mapping crucial genomes. So there are different disadvantages and advantages. Let's see the advantages first. It is less degraded compared with protein and mRNA. DNA is very stable. It is efficient way to detect specific DNA sequence from large complex samples. Increased sensitivity of fragment detection because of the probe labeled using amplifying signals also helps in more prompt detection. It is only way to diagnose FSHD disease, that is fascicular pleuromolar muscular dystrophy. This is a disease that can be exclusively detected only by Southern blotting. There are certain disadvantages also. Let's see the disadvantage. It is not applicable in routine diagnostic settings. It's time consuming and requires large amount of DNA also. Does not allow morphological preservation of tissue. You cannot preserve the sample here. So historic evaluation feature are not available, means once the test is done you cannot save that test. So that is the disadvantage of this technique. But still this technique is a very important technique and there is no replacement to this technique till date. Thanks for watching. Please press like button if you like the video, share, comment and subscribe the channel. Thanks for watching.